So the last talk will take us all the way from theory to practice, and there's no better person in the room to take us all this way, and this done up here. Right.
Is this on? Okay. Okay. So, as you can see, I changed the title. And Aviv promised that I'll discuss all sorts of things. The number of minutes I have isn't enough. And a disclaimer, most of these slides were prepared with after lots of glasses of wine with John Marioni. Oh, this is not... These aren't my slides. Someone changed my slides behind my back, which is great. This is going to be a really interesting talk. So this is, from Marit's slide earlier, and here the goal is, yes, we want to have this atlas, this thing which we haven't described, which, we can do reproducible, and if we take it from another person, we could just, slip it in there. But, I actually think that there's a problem with this definition when we're thinking about 1.0. And that's what we're thinking about here, because, our funders are very generous, but we really don't have the type of funding we need to get to the end goal, so we need to make 1.0 meaningful. So I really think the definition should be, now this is stuck with this big X. The definition should be, how can we design this 1.0 to be useful? And since we're having breakouts about designing and building a roadmap for the different organs, this is by no means comprehensive. I don't promise it will be even coherent, but I'm hoping to give you some food for thought on how do we get to an atlas that's somewhat meaningful. Oh, I know what that question mark is. The graphics got rendered bad, but okay. Never mind. It made it quite interesting. So, in this world of single-cell technologies, this is sort of how I view most of us. We have technologies, and we're excited like a kid in a candy shop, and out comes new technologies, and then these new technologies make us excited again. But, I want us to stop and think. And beyond an atlas, we really want to say, well, what do we want to use this atlas for? How can we build a roadmap? How can we plan an atlas if we don't have what are our questions? What are our queries? What do we want to use this atlas for? How are we going to use it? Because if we want to build a roadmap and make a plan, we should have clearly stated goals, clearly stated questions. And I was sort of pushing on my friends in the lung atlas community, and I told us, what are your questions? What are your questions? And in this group, there's a bunch of people, and they're all interested in different diseases. It's sort of what somewhat allows them to play nice. It's not a complete overlap between the diseases, so it's a little easier for them to play nice. And so we decided that we were going to really focus on a couple of regions that play really important roles in diseases, and people sort of sat down and say, here are the regions that have, the biggest role in these diseases that interest us, and that really also defined the goal of, we want the healthy human lung so that we can compare to and understand what goes wrong in disease. That's already some, inkling of a query. That's some inkling of a goal. And so, okay, if that's our goal, let's think about it a little bit. And again, this is just, early thoughts. Here's some data generated by Pascal Barbie. And he compared two types of biopsy techniques, brushing and forceps, inside the nasal cavity, and actually found that there are big differences in the cell composition, particularly in the depth. So if you have healthy cell types from one type of sampling techniques and then try to compare a disease with another type of sampling disease, you might get compositional differences like we see here. And Naftali Kaminsky, he's really interested in pulmonary fibrosis, so he built a pretty deep atlas with pulmonary fibrosis, about 90 different samples of both healthy and diseased. And what he found is actually that most of the cell types you see in fibrosis and in diseased patients, you see them in healthy patients. There's no big new cell type. It's all a matter of their proportions, their composition. There's just a huge change in the composition when you go from these sort of control, which is healthy, over to the different type of fibrotic patients in terms of their epithelial compartment. This is not to say that there's no new cell types that come about in fibrotic patients and pulmonary fibrotic, but the new cells that come about, and they seem to, we might even see them in normal, they're really rare. They're about 5 in 10,000. So if you take a typical sample size from a patient, you're unlikely going to find it. So if you really want to understand disease to normal, then a lot of it is going to be in the composition, in the histology. So when you're designing what type of sampling you're going to do for the healthy with these type of queries in mind, then you have to actually think about, okay, what type of sample sources am I going to have from disease? Because I'm going to compare these things to normal. And so here's, Aviv's slides and the different things that people might want to query. And so, at the level of queries, the first one, one wants to query and say, hey, just, here's my sample, annotate it for me. Something like, super version of Peter's annotation engine. What do I have here? Here's my data, find all my programs, my metacells, my cell types, my histo-modules, and everything else I can get from the sample. The other level is more, the next two queries are more, do we understand? One thing we can ask about our atlas is if we get some type of samples, for example, just a bunch of disassociated cells from single-cell RNA-seq, do we know enough? Is there enough connections between these modalities? So if I just have a population of cells, and I assume it's healthy, I can figure out how these cells come together in these proportions into structural organization, into histo-modules, where they might be in the organ. Do I have enough information for doing that just by collecting a bunch of disassociated cells from an unknown region? Of course, this is just testing what we know, because usually we'll sort of, if we're good scientists, know where those cells came from. And finally, for the disease case, is there any deviation? Deviation in the cell types we see, deviation in the programs and in the genes we see them, deviation in the composition, in the fractions, and in their spatial organization into histo-modules. And so, again, many diseases are going to be at the level of, doesn't really work, at the level of spatial organization and fractions. We've seen that in a couple cases, and we really need to take this into account and take into account the similarity metric so that we'll be powered to ask the questions that we want. Now here we have, I think the battery here is dead. Here we have a comparison between single-cell RNA-seq and single-nucleus RNA-seq in the lung. And one might think this is terrible, like they're completely different things. But if I didn't show you those maps of them t-SNE together, then actually if you cluster each one of them separately, and you annotate them based on the way we like to annotate things in the future using Peter's tool, you'd actually find all the cell types you expect with all the markers you'd expect, and you'd never know that those two data modalities don't, sit upon each other so much. So how you collect your data, and again, how your query samples are going to look like, what type of technologies are you going to have available. For example, pathological samples are often biobanked and will often need to be done at the nuclear level, so you need to sample enough of those and understand these differences. Of course, one can say that we can magically fix it up together. There's John Marioni's MNN correct. It's a great thing. Here's a case where you take different, this is pancreas, different data from different technologies, and you put it all together, and it's perfect. But is it perfect? Here's some data that we collected in AML. I like to study cancer. And at first, you see every single patient is completely at a different place. Huge batch effect. I messed up. I always mess up. But when you dig into it, you see that actually, if I look at the healthy cells, the T cells, the B cells, and the erythroids, they're all really mixed up. The cancers are really, really different. So all those, reviewers who always want me to do batch correction and smush my cancer samples that are different because they are different cancers together, that's a mistake. Keep in mind, these batch correction methods, all of them, not only John's, make the very strong assumption of these are supposed to be the same, and I'm going to force the data to be the same. And in many cases, the data isn't the same. If I want to compare, for example, macrophages in the lung and the gut, and I'll try some type of correction method, batch correction method, I will make them the same and obscure biological differences. Likewise in disease, or if, for example, we want to compare if our in vitro organoid is matching in vivo biology and where it differs. So these batch correction methods, they're not quite ready to distinguish between differences that we see because of technical noise and because of true biological differences. And I know that a lot of people in this consortium think that some magical computational elves will take all this different data that we're collecting with different modalities and fix it and turn it into one atlas. And I can tell you that I don't know such computational elves. We're still grappling, and we're going to have a meeting in November about normalization of a single sample. It's normalized across data modalities and collection techniques and how the sample was handling. That's very far in the future. So we're going to have to take that into account when we're designing the atlas, and that's why Aviv and I started to think about, okay, the cells, they're data, they're messy, they're dirty. Let's try and think of abstract entities, of entities that we can identify in our data, and then we can compare these entities. And that's, I'm not going to describe all of them, but we have like cell types and states that live on trajectories and dynamic transitions between them, and have rules, they have modules, they're spatially organized, they're collected from somewhere. And if we have an abstract language, if we can maybe not normalize all the cells to map onto each other in a perfect, whether it's a t-SNE or a UMAP or force-directed layout, if there's some type of difference due to batch, it's going to come across in any one of these visualization technologies. But just like I said, that at the end of the day, when we clustered and we got the cell types, we did get a match of the biology we expected. So let's try and dig into this. I'm going to talk about dendritic cells. There's some people in the audience that love them. And here's a tabulamuris, and you see that these guys are really rare. You could see one or two little green cells. So with Sasser-Densky, we decided to dig into them. This is in mouse, and we did FACS sorting and actually found a pretty rich clustering of dendritic cells. So here's our, labeling, our annotated cell types. That's actually different than clustering. Clustering and annotation isn't the same thing. And also, different clustering algorithms sometimes give you different answers, and that's where you actually need a domain expert to come in and say, this is what defines the cell type, this is our ontology, this is what it means, this has the functional definition. Now, again, clusters are not perfectly defined because they differ depending on your algorithm. But if you actually look at the data you collected, the data has density. So this is the same data and plotted as a contour map. And what we see is their real structure here. And I'm going to zoom in even further and show that this data, and we've seen this reproducibly, whether you map it with t-SNE or UMAP, there are these density peaks in the data. And they come up, and you can sort of zoom in more and more and get denser peaks. And that density peak is what we started defining, and it's not a final definition of what a metacell is. This cell is some type of biological condition which is built on some type of different programs that biologically matter. This is some type of state the cell can be in. That's a data-derived definition. And everything around it is just like some white noise, whether it's biological noise of genes that matter less for what the cell's trying to do right now, or technical noise in our measurement, but really what we have in the data, and I've seen this in almost any data I've looked at, are these very distinct peaks. And these very distinct peaks are defined by the activation of different gene expression programs. Different genes are expressed, and the different genes are expressed in different covariate structures with each one of these programs. So if we take a first stab at sort of defining what a cell state is, what a cell type is, a cell type is, a cell type is sort of more refined. Then there's this real data-driven density here. And so a cell type is a functional annotation. Here we need a collaboration between the data and the biologist to say, this is a type, it's a static thing, we know what it means, we defined it together, we agreed upon it, it's a thing. And a metacell is a data-driven definition. We see these density peaks in the data, we collect data, we still haven't interpreted it, but it's a very clear signal in the data which we could define in mathematical and statistical terms. I wanted to go in and delve a little bit more into programs and how they can dynamically change. Here is bone marrow development from another data set. And here you can really see what programs are. You can see that over time during erythroid development, you have different transcription factors that come on in waves. And along with this transcription factors come entire programs of co-regulated genes that come on and change over time. And over time, these have very, very different covariate structures. And I know people don't like the concept of covariance. They think that mean behavior summarizes everything, but there's actually a lot of information in covariate structures. Here we have some data that we collected in TILs from breast cancers, as well as some healthy breasts. And what we see is that particularly many of the cell types like Tregs or macrophages, we actually see that many times what distinguishes between the clusters are their covariate structures. And you can see very, very different clusters of co-expressed genes that go together in very different ways across the clusters. And why I think covariation is such an important concept for us to be thinking about is that we collected this data using 3 technologies. We collected this data using, old 3' indrops at the very beginning with a 30-minute ride between at Memorial Sloan Kettering to Columbia. We collected this data with 5', 10x, freshly from the operating room, and with CyTOF. And there were big differences between the technologies, but the covariant structures of the clusters mapped one-to-one perfectly with no effort whatsoever, no normalization, nothing fancy. Covariate seems to be something very strong in the data. So again, a data-driven, this is sort of cut versus a biological. A gene program is, again, something that we can annotate and we understand. A bunch of genes that are doing something together in a biological function that we can call by its name and what it's doing and why they're working together. And a metaprogram is, again, a data-driven statistical feature of a covariate pattern of genes that changes in an interesting way across our metacells and cell types and things like that. So how do we build a roadmap? Again, I cannot stress enough. You have to stop and before you jump into how many samples and how many individuals, what do we want to get? What do we want to achieve from version 1.0? You have to stop and think and answer this question. And when you have this question, the AWG would really love to know. And yes, please, send us your stuff. What do you need to capture for your question? What type of individuals do you need? I'll tell you a little anecdote. When I was at the beginning of my faculty, I was looking at TCGA ovarian cancer. And the control set was very thoughtful. Someone really thought it through to give the easy biological result of 100% ovarian cancer patients have a deletion in their Y chromosome and a duplication in their X. That was very thoughtful to have all the controls that way. Anatomical and histological reasons. We're not going to be able to do all of them, so choose carefully to be able to show value. If we want to go to the next stages, we have to show the value of phase 1. What rarity of cells do you care about? Some of the cells I showed you, such as the dendritic cells, are very rare, yet they play important roles, for example, in type 1 response, and include exploration. We don't know everything, so we should do some random stuff just to find something by chance, because remember, we want to use this to plan our next step. So there's tracking metrics. I'm almost done. There's tracking metrics, and that's sort of the bean counting. I know that many of, for example, my NIH officer really cares about how many samples I did, but really, I think utility metrics. How close are we to our goal? Not only how many new entities are we discovering, but at what rate? Are we still discovering more? How close are we towards successful querying of the things that we want to know? Of course, we have to define our queries in advance, define good similarity metrics, and define good metrics for success of these queries to track that. All these things that is homework for you to define. Deviations. Again, how well can we model deviations from the normal? Again, important to define these deviations, what we want to achieve, how accurate and with what metrics. And this really should serve as information for the next phase. So we welcome your ideas and input. I hope that a lot of people will think about types of queries, other abstract entities, or better definitions for abstract entities of interest that we could define inside the atlas, as well as utility metrics. So there's now going to be a breakout, which other people will define, but I hope that people think about these things and share with us.
Yeah, thank you very much, Dana, also for reminding us to pose proper questions and to remind us to have the questions.
Yeah, I think we once upon a time learned to do that when we didn't have such data-rich technologies.
You had more time to think about it. Okay, so there's a question in the back, please.
Dash to the past, about 2005, Sui Wang and talking about attractors.
Mm-hmm.
So as an abstract concept, it kind of looks like your peaks are kind of like either attractors with noise or attractors that actually have biological, the noise is biological in meaning. So going back to your idea of abstract concepts, is that something you've been thinking about? Because it goes to the idea of like states and types being segregated in a way, even though, I mean, we're mentally segregating them, but they're really maybe not segregated.
So attractors, I think, are a subset of what we're looking at, mm-hmm. and therefore I don't like that terminology because it assumes some type of dynamics. The way I think of these, first of all, again, my metacells are supposed to be defined by the data, and there could be different reasons that I see these peaks. Some of them are true measurement noise. This is really noisy data. It could be that all the cells are completely identical, probably not, but all we see is measurement noise, and that's some of that. Additionally, some of these states, and they're not necessarily an attraction state, they're just representative of where the cell is, what its environment is, what signals it's getting. There's some genes that are very noisy but are actually at low levels of expression. We did some tests where we put some modules and then we put genes at extremely low level of expression, really the level of sort of leaky noise. And again, we see that same shape because even though it's very, very low expression, very, very noisy, it's just a lot of genes, and that creates, again, these peaks. So, the attractors is some type of dynamic process. And here I just think that we have these states that really represent coherent biological programs of something biological going on, the cell receiving a signal, responding to its environment, doing something, screaming, I'm a cell of this type, and here are my canonical markers. And those, you have a bunch of coherent genes that give you the signal in a really strong way. And the other things that don't have to do with the biology that's going on right now in the cell sort of create white noise around it. And I mean, that's my current thought. I could be completely wrong.
That's a very good statement. But okay, Fabian, a very brief question, please.
Dana, John and I were a bit confused what the tetrahedron here would mean in terms of, like, how would you really computationally describe? Isn't that just a set of features that you now describe for each cell? Why should they add up or be sort of lower than in the sum than 1, which is sort of indicated by the tetrahedron?
So the tetrahedron is basically meant to represent the idea that there are many different facets, that they're connected, and what we hope to do is actually gain understanding in these connections, so we could predict some of them. We could look at a subset of these projections and predict the other ones. We can even understand the underlying mechanisms that make these. So the tetrahedron really is just a thing about there are different facets to a cell. We measure some facets using some technologies, and if we really understand the system, then we'd be able to connect and see how they all connect together. And that's one of our goals, I hope and believe.
Okay, the final question goes to the...
Here. So on the point of data integration and also our choice of kind of cases and controls, there's two main ways that you can kind of approach it. One is where we build our healthy atlas, and then we build our disease atlas, and then we try to pattern match, and you kind of raise some of the issues around that. And then there's the ways that practically we usually do it. We're all excited to analyze our data early, and as we collect both, we kind of build our atlas simultaneously of health and disease. leading to various issues in both situations. Do you currently have any thoughts on kind of best practices of building separate trees and then matching, or building a consensus tree together and then kind of trying to extract back out what was really healthy and what was really disease?
So, as you said, everything has problems because we don't completely understand the data. I think that the planning for these two things should be together. So we should keep in mind, as I said, the queries that we're going to want to do with this atlas and the type of sampling techniques and technologies that will be possible for a disease sample. It won't help if we do all our atlas using data from biopsies and fresh cells, and then do all our disease studies with single nucleus in resected samples. And I do think that at the end of the day, it's very hard to map things together. That's what I showed. And therefore, I think that whether we collect the data together and collect the data separately, and we should always have our roadmap and our experimental design to make the least amount of confining bias possible, that yes, we are going to have to think about these abstract entities that make comparison better. We are going to have to think of similarity metrics. I think the idea of just doing some type of normalization and batch correction and harmonization that we don't completely understand, and just say, here's how the TSNE or the UMAP overlaps and doesn't overlaps, and here a region where it doesn't, can lead to horrible artifacts. And therefore, this abstraction is a very important step no matter what you do.
You can also let my reviewers know the next time.
What?
So in any case, thank you very much again, Dana, and for everybody contributing to the discussion, to all speakers. So I need a break, definitely. The organizers donated us 5 minutes additional time, so we're going to start with the breakout sessions at 3:30. So if you are hungry or you're up for a coffee, you'd better run out and resume in 50 minutes. Thank you.
Let me quickly guide you to the breakout session rooms.
Yeah.
So we have 6 breakout sessions in 6 breakout session rooms. The auditorium will be here. Then you have the alpha, beta, gamma is leaving the auditorium. Then straight to the left, the agora. The easiest way to go through this door, or follow a line that says agora, and then down with the elevator. And the tau is very easy to find next to the registration desks. Thank you.